Thank you very much for joining Life Science Exhibit Virtual Conference. My name is Debayan Dey. I am a Cystic Fibrosis Foundation Postdoctoral Fellow in the Department of Biochemistry at Emory University School of Medicine. I'll be discussing today our research on interaction of respiratory pathogen Pseudomonas aeruginosa with human epithelial cell. This is the outline of the webinar. I'll be starting by giving an introduction to the pathogens and the global threat. I'll explore some of the previous work done on phosphorylcholine and its mimics in host adhesion, some of the previous work by Goldberg and Conn labs at Emory University on understanding the Pseudomonas host cell adhesion by COP mimicry. And then I'll be discussing my work on substrate recognition by Pseudomonas aeruginosa EFTU methyltransferase EFTM, which is published a few months back. At the end, we can have a question and answer and discussion session. Now let's talk about bacterial and viral respiratory pathogens. As we see here, respiratory infectious diseases are generally caused by bacteria and viruses which infect the respiratory system of humans. The upper respiratory tract hosts a large number of potential pathogenic bacteria and commensals, making a complex microbial community. We see here in the slide Pseudomonas aeruginosa, Haemophilus influenza, Streptococcus pneumoniae, and many other pathogens are present in the respiratory tract. On the other hand, respiratory viruses, as you see here, rhinoviruses, which cause the common cold, RSV causes respiratory tract infections, and the coronavirus family members like SARS-related coronavirus or the novel coronavirus SARS-CoV-2. It causes severe respiratory infections at present, which is the cause of a current ongoing pandemic. Now let us look at the statistics. Among the top 10 causes of death worldwide, the lower respiratory tract infection and tuberculosis combined kills more than 2.5 million people each year. This is a combined death from the bacterial and viral infections. Now, as we see and look at the data for the pneumonia-related deaths, also both caused by bacteria and viruses, it kills more than 1 million people. And it has a high fatality rate in the developing countries, as you see here in Africa, the Indian subcontinent, and also in South America. In US, the seasonal flu infects more than 9 million people each year, with 140,000 hospitalization and 12,000 deaths each year. And this is a very conservative estimate. If you see the upper limits for this, which is the 2017 and 18 season, total number of people died due to flu is 60,000, which is actually huge. Now let's a little bit talk about the bacterial and viral collaboration in the respiratory epithelial surface. The bacterial and viral synergism is well studied, and this collaboration in the respiratory epithelial cell is one of the major causes of mortality in the co-infected patients. The synergy between Streptococcus pneumonia and influenza virus is a textbook example and studied for more than 20 and 30 years. In this figure is human lung epithelial cell, which covers the surface of the airways and alveoli, are the first targets of the exposure of inhaled substances, and it is the first gateway for interaction with viruses and bacteria. I'm going to show here a simplistic model in which when virus interacts with the human epithelial cells, what happens. So in the simplistic model, the virus interacts with the epithelial cell and starts a cascade of reactions in which the first is ciliary damage, and there is decreased mucociliary function, and there is a rupture of epithelial integrity. They also reprogram the host cell to upregulate a broad range of receptors and protein signals. Among them shown here is platelet activating factor receptor PAFr and receptor for sialic acid RSA. But there are other important receptors are also been upregulated. This whole mechanism further encourages the bacterial invasion and their adherence, which we'll be discussing in a moment. So now let's talk about antibiotic resistance. As we now find ourselves in a pandemic caused by SARS-CoV-2, the novel coronavirus, which has killed around 250,000 people worldwide, there is another pandemic which is slowly going on in the background, and that is antibiotic resistance. Now, what is it? Antibiotic resistance happens when bacteria develops the ability to defeat the drugs which is designed to kill them. Infections caused by antibiotic resistance pathogens are very difficult and sometimes impossible to treat. Many medical interventions are dependent on using antibiotics, including organ transplants, cancer therapy, and treatment of chronic diseases. So there is always a tug of war going on with the drug and the new antibiotics. And the bacteria always is evolving new and new molecular mechanisms to make these drugs ineffective. As you see here in the data from CDC about the situation of antibiotic resistance in US, it kills around 50,000 people each year. It's a staggering number, and it infects more than 3 million. In Asia and Africa, the death per year is nearly 1 million currently per year. Again, a huge number. If this trend continues and efforts are not made from public health, regulatory, and research standpoint, we could easily see 10 million deaths each year by 2050. Or it could be sooner, who knows. Now, as we were discussing in our previous slides, that there exists a synergy between the viral and bacterial infections. It is becoming more visual nowadays with the COVID-19 and antibiotic resistance both in play. Preliminary research has shown that patients are more vulnerable to superbugs when infected with viruses like SARS-CoV-2 and their immune system being weakened. In 2009 H1N1 influenza pandemic, it was shown that more than 29 to 55% of deaths were caused by secondary bacterial infection. It is like the viral infection weakens the immune system and facilitates the bacterial invasion. And as the greater percentage of bacteria, as we know in the hospital setting, are antibiotic resistance, treating them along with the virus is becoming a very much big challenging deal. And this is not new. Studies conducted by Anthony Fauci et al. In 2008 has shown that the vast majority of 100 million deaths in the Spanish flu pandemic that happened around 100 years ago, 1918 to 1919, were caused not by the viral influenza alone, but by the respiratory secondary bacterial infection which invaded the lung tissue, which has already been ravaged by the flu virus. Well, as we now know that many of the antibiotics like azithromycin, amoxicillin, tetracycline, doxycycline are being used in the hospital. They have been experimented with to treat patients. There is actually a growing risk of, again, a surge of antibiotic resistant strains getting produced in this hospital in the coming 2-3 years. So what we need to do? The urgent need is to strengthen cross-disciplinary research, develop strategies to contain these respiratory viruses, of course, with vaccines and antivirals, which the pursuit is going on in the world. But also there is a greater need to redesign the antibiotics which can evade resistant mechanism. Now let us discuss about bacterial adhesion to host cells. So the adhesion to host cells using cell surface associated protein or secretory machineries is very crucial and requisite for the bacteria to engage various machineries of virulence factor to impact the host cells. Gram-negative bacteria use secretory and toxic injecting machineries like the well-known type 3, type 4, and type 6 systems to inject effector proteins to modulate or reprogram the host cellular machinery. Proteins are injected in a programmed manner into the host cell, and if the bacterial host adhesion is not strong, the translocation of proteins are disrupted prematurely and the infection is not effective. Thus, there is a great need to understand the biology of bacterial and host adhesion, and the recognition of different pathogens is crucial for designing new anti-infection strategies. Thus, what we can do is, using structure-guided or high-throughput methods, we can design anti-adhesion molecules which can inhibit cell-cell adhesion by mimicking the bacterial or host cell binding partners. This is an alternative strategy in which antibodies can be made which can recognize bacterial surface epitopes and can immunize the host. Now let us discuss about phosphorylcholine and host adhesion. So phosphorylcholine is composed of a negatively charged phosphate bonded to a small positively charged choline group, and is usually a part of some phospholipids. So phosphorylcholine will be referred to COP for choline phosphate from now on. So COP is a part of recognition domain of host protein platelet factors, PAF, which binds to host platelet activating factor receptor, PAFr, on the epithelial cells, as shown here in the figure. Now, in the same COP is actually utilized by the bacteria to mimic host cells and adhere to the epithelial cells. As shown here, the Streptococcus pneumoniae with the extracellular COP, it competes with the host cell and then interact with the PAFr in the epithelial cell. This is the mechanism by which the PAFr binding increases cell invasion for different bacterial species, including Streptococcus pneumoniae, Pseudomonas aeruginosa, Haemophilus influenza. The invasion of epithelial cell also allows the bacteria to avoid extracellular immune responses. Now, the bacteria can use this also to modify host immune response. Now, well, the empire also strikes back. That is, the host can also respond to this mechanism. In Haemophilus influenza, acute phase reactive C protein, CRP, recognizes COP and can start complement-mediated killing and can also start phagocytosis. So I can now want to focus more on Pseudomonas aeruginosa and epithelial cell interaction. Mostly the Pseudomonas aeruginosa and the epithelial cell interaction is studied by the groups of Goldberg and Conn. So previously, the labs of my mentor Joanna Goldberg and Graeme Conn has explored the role of COP mimics in Pseudomonas adhesion to the lung epithelial cells. So the first 4 papers where actually they showed how this whole thing happens. This next paper of Kuiper and Dey et al. Published just a few months ago actually shows the next phase where we wanted to understand the whole mechanism from a molecular point of view. And the question is why we want to study Pseudomonas, why Pseudomonas is important. Pseudomonas is an opportunistic pathogen which causes life-threatening acute infection in individuals with compromised immune system. It is also an extraordinary antibiotic-resistant bacteria. It is a common cause of chronic respiratory infection and one of the leading cause of mortality in the patient with genetic disease cystic fibrosis. In the 90s, Joanna Goldberg and team has shown the presence of COP or a COP mimic on an unidentified 43 kilodalton protein on 12 Pseudomonas aeruginosa clinical isolates. That time it was not very clear that whether the modification contributed to the Pseudomonas' ability to colonize the airway tract infection with PAFr, because the expression of the COP was directed only at the lower temperature, that is around 25 to 33 degrees centigrade, but it is absent in the 37 degree physiological temperature. The work was continued later in 2008 from Joanna Goldberg's lab. So Barber et al. Has shown that the Pseudomonas aeruginosa strains from chronic infections are also expressing COP at 37 degrees. Here the author has shown in a representative Western blot that the COP epitope expressing using an anti-COP antibody in Pseudomonas aeruginosa acute and chronic airway isolates were grown in 22 degrees and 37 degrees. As you see in the figure, the acute infection, there is a COP present at 22 degrees, but is absent at 37 degrees. But in chronic infection isolate, it was found that at both 22 degrees and a little less in 37 degrees, the COP is present. They also determined the identity of the 43 kilodalton COP containing protein, so where COP is expressed. So the authors subjected the whole cell extract of Pseudomonas of PA01 lab strain to a 2D gel analysis. They excised the spots and then subjected it to mass spec, and they found that the 43 kilodalton protein correspond to the translation factor EFTU. We'll be discussing about EFTU in the next slide. The author also showed that the dose-dependent inhibition of adherence and invasion happens against the PAFr antagonist. As you see here in the figure, that as the concentration of the PAFr antagonist is increased, you see less and less cell adherence and also less invasion. That tells that the EFTU modified with the COP or a COP mimic is actually disrupted. The interaction is disrupted with PAFr, and overall the invasion and adherence is decreased. Now everything is happening in EFTU. So what is this protein? So let us discuss about the canonical and the moonlighting function of this interesting protein. Now let me introduce you to this protein EFTU. It's an essential and universally conserved GTPase that ensures translational accuracy by catalyzing the reaction that adds correct amino acids to a growing polypeptide chain. After the incoming amino acyl tRNA docks with the mRNA, the GTPase activity induces a conformational change which releases the EFTU from ribosome. EFTU, it has three domains as shown here in the figure, domain 1, 2, and 3. The domain 1 is the N-terminal domain where you have the N-terminal first 10 residue flexible peptide also. It is also one of the most abundant proteins in bacteria and often associated with cell membrane, not only in Pseudomonas, but also in many other organisms. So the interesting thing here is EFTU lacks a signal secretion motif, doesn't have a signal peptide, but still it can reach the cell surface. Still, this is not known. We don't know how, and this is again a matter of investigation by our groups. It has a multifunctional adhesive characteristics and it plays a crucial role in host pathogen interaction as shown in many organisms. So that's why we are very interested to study that how the EFTU and its modification with COP helps in its role as an adhesin to the human epithelial cells. To further this quest, Goldberg lab tried to identify the genes responsible for post-translational modification of EFTU. They screened by Western blot analysis a select set of transposon mutant library for genes which were previously known or potentially involved in the synthesis of COP that has been identified in other bacteria. But to their surprise, none of the Pseudomonas transposon mutants which they tested showed any altered COP expression. That tells that there may be a novel pathway working to add the COP to EFTU, or maybe there is something else, maybe there is a COP mimic. So to understand that, the authors conducted a systematic analysis of the transposon mutant library in Pseudomonas aeruginosa PA14 cells and utilized the whole cell lysates of more than 5,000 PA14 mutants to check for the loss of EFTU modification, and they probed it by anti-COP antibody by Western blotting. They found, as you show in this figure, that there's one mutant which codes for a novel class 1 methyltransferase, and this mutant did not show any reactivity either at 25 degrees or 37 degrees centigrade, as shown in this figure. The same thing they observed that in PaO1 there is COP reactivity by anti-COP antibodies at 25 degrees centigrade, but there is nothing at the 37 degrees centigrade. But then if they again delete this gene, which they termed as EFTM, which means EFTU modifying enzyme, they again found that it loses the COP reactivity both at 25 and 37 degrees centigrade. So the next thing what they did is in the delta EFTM strain, they complemented the EFTM and they got back the activity again, this time at 25 and 37 degrees centigrade. So now the authors suspected that the methylation in EFTU may be responsible for adhesion and not the COP, because there's a similarity in the epitope of the trimethyl lysine and COP. So now they wanted to find whether really the trimethylation happens or a dimethylation happens in EFTU. They wanted to know what is the site and the nature of methylation. So they did mass spectroscopic analysis, and which showed that lysine 5 is the site where trimethylation happens. To prove it, they created a EFTU lysine 5 to alanine mutant, and the results showed that this mutant does not react with either of anti-COP or anti-ditrimethyl antibody. Now in oropharyngeal and bronchoepithelial cells, it was shown that purified trimethylated EFTU binds better to host cell compared to an unmethylated host cell, as shown in this figure. Also, it is better internalized. So it tells finally that the trimethylation of EFTU lysine 5 represents a novel bacterial strategy to mimic the structure of a natural ligand of PAFr. The authors' finding also indicated that the trimethylation of EFTU mediates the interaction of Pseudomonas aeruginosa with human epithelial cell via PAFr, but it does not confer increased susceptibility to the bactericidal effects of the complement system. It also suggests that the trimethylation is different from COP in some aspects of bacterial host interaction, and this is something we need to investigate further. Now the question persisted that why the modification in acute infection isolates is lost at the physiological temperature of 37 degrees. And what happens to the EFTU methylation at the higher temperature? So here Conn lab comes in the picture, and it collaborated with Goldberg lab to study the mechanism with biochemical and structural expertise. Now Conn lab has an extended experience in studying methyltransferases from pathogenic organisms majorly involved in antibiotic resistance. The first objective is to clone a recombinant EFTM, express it in E. coli, and to see whether EFTM is really an active methyltransferase. Through the screening of clinical isolates, the author has identified that the strain PA-HM23 has failed to methylate the EFTU at either temperature, both 25 and 37, as shown over here. The sequencing results show that there's a single amino acid substitution G50R. So there's a glycine to arginine mutation, which correspond to the putative SAM binding site. SAM is the methyl donor. Using an ITC, as shown over here, it shows that the wild type and the G50R mutant, and as we show here that the wild type can bind with SAM with a great affinity, but the G50R couldn't bind. So that tells that our EFTM is an active methyltransferase, and with the single mutation in the SAM binding site actually disrupts it. Now here the authors show that EFTM is necessary and sufficient to methylate EFTU without any other protein factors and with just SAM available to it. In an in vitro methylation assay shown here proves that at 25 degree, clearly shown here, at 25 degree at 0, 5, 10 and 20 minute time interval, it can do the reaction and we can see the modification of EFTU. But at 37 degrees centigrade, just after 5 minutes, clearly shown over here, the reaction stops. So that means there is no more modification on the EFTU that is made. So the author wanted to investigate further using circular dichordism spectroscopy whether the temperature was affecting the enzyme secondary structure, that the EFTM secondary structure, which might account for this temperature sensitivity. Now, as we see here at 25 degrees centigrade, the CD spectrum of the recombinant EFTM is consistent with a well-folded protein. Now, when the CD signal is fixed at 280 nanometers and we collected a linear temperature gradient, the EFTM structure remained stably folded over 20 to 30 degrees, but subsequently started unfolding at high temperature. Using another complementary technique, which is known as differential scanning fluorimetry, it also showed the same result, that after 30 degrees centigrade, it slowly started unfolding. This solves the mystery of the thermoregulation of EFTU modification at 37 degree and show that there is a new way of thermoregulation of EFTU by this novel methyltransferase EFTM. Now, to recap what we have learned so far, in Pseudomonas aeruginosa, EFTU doing its canonical role as shown here. It is binding to tRNA and playing a role in the translation at environmental temperature 25 degrees centigrade. But as we know that EFTU is also a very abundant protein, so it is present all around the cell. It also is exported outside the cell and presented itself outside the cell. Now, EFTM promoter, as you can see here, creates EFTM protein. The EFTM protein at this 25 degrees centigrade, at that time it is stable. It can interact with the free EFTU and can methylate in the trimethylate EFTU at the K5 position. These methylated trimethylated EFTU can go by an unknown mechanism to the cell surface as shown here. Now at higher temperature, what happens is the EFTM promoter is switched off and the EFTM is also thermally degraded. But still there is enough EFTU trimethylated which is on the cell surface, which is attracted by the lung epithelial cell, and there is an interaction of the trimethyl lysine moiety of the EFTU with the PAFr in the lung epithelial cell as shown over here. And this is the reason for the bacterial cell adhesion or the Pseudomonas cell adhesion to the lung epithelial cell. Now, we wanted to know a more detailed molecular mechanism. And this is where I joined the Conn lab in 2017 and started working on the next phase of the problem. My bioinformatics and evolution analysis suggested methyltransferase EFTM has a class 1 methyltransferase fold, as it was known earlier, but the structural elements still resemble set domain methyltransferases found in eukaryotic histone modification protein. And this is not at all understood. Also, our analysis showed that the mechanism of interaction may be different compared to the known class 1 methyltransferases in bacteria and also from the set methyltransferases. In general, the interaction and the mechanism of action of lysine methyltransferases in prokaryotes are very much understudied. So we wanted to define the EFTU-EFTM interaction as a first step in understanding the molecular details of trimethylation and then EFTU-mediated cell adhesion to the host PAFr. In the next step, we want to understand the transport of EFTU to the cell surface, its presentation and anchoring in the outer membrane, and ultimately its interaction with PAFr. Now, why we want to do this? We want to understand the molecular details because it will assist us in designing inhibitors to target EFTM, which is a very prominent drug target in Pseudomonas. Now, this inhibitor could synergistically act with other frontline antibiotics to arrest the adhesion and also to kill the pathogen. So in that, I'm going to discuss our paper which published a few months ago about the first phase, which is understanding the substrate recognition by Pseudomonas aeruginosa EFTU methyltransferase EFTM. Here we show a homology model of EFTM. We are trying to get the structure of EFTM by crystallographic method. For the time, we resorted to generating a homology model. And you see the structural elements over here. It has a class 1 Rossmann-like fold, which is shown in gray over here. It has a SAM binding pocket, very conserved among all the class 1 methyltransferases. The uniqueness of this methyltransferases and related family of methyltransferases are the auxiliary domain, which is the beta 5, beta 6 linker. You can see here in this, which is the blue color region over here. So now this is the extra domain added into the methyltransferase fold. Now in Pseudomonas aeruginosa isolates and laboratory strain PA01, EFTU modification is present in bacteria cultured at 25, but is absent in 37. EFTU methylation was previously observed in 37 degree only in chronic infection strains of PAHM4. Thus, we wanted to investigate this particular EFTM homologues, folding and functional properties. And the reason we wanted to know, because we wanted to have a stable variant or stable homologue of EFTM for our biochemical studies, because of the inherent stability will help us for doing structural and biochemical studies. Using differential scanning fluorimetry, again, we observe that the melting temperature of EFTM from HM4 is increased by 5 degrees to 37 degrees compared to 31.5 degrees of EFTM PO1, as shown in this figure. The black line indicates the EFTM from PO1 and the red line indicates the EFTM from HM4, and you can clearly see a shift in the melting temperature and it becomes a little bit more thermostable. Now next we did a functional analysis or the activity analysis of HM4, and we observed that HM4 retained the enzymatic activity after incubation at either 25 degrees or 37 degrees centigrade, but its activity is ablated after incubation at 42. It actually shows that EFTM from PO1 and EFTM from HM4 are functionally equivalent enzyme, and the EFTM from HM4 exhibits thermoregulation but shifted to a higher temperature. To begin defining the EFTU recognition by EFTM, we first asked whether important interactions are made with the surface of the globular protein of EFTU, or if isolated N-terminal peptide alone can make a suitable substrate for the modification. Now, the reason for the question is many lysine methyltransferases of set domain found in eukaryotes can recognize just the flexible peptide alone where the modification has to be made. So we wanted to know whether the whole body of the protein is required or just the peptide is enough. We compared the ability of EFTM to form a stable complex with EFTU and a delta 11 EFTU, which is the variant in which the unstructured region part containing the lysine 5, where the modification is made, is deleted. And what we observed using gel filtration chromatography, that EFTM is able to bind to both the wild type EFTU and the truncated EFTU nearly with the same affinity. We want to show the same thing using isothermal titration calorimetry, and what we show here is the wild type EFTU and the delta 11 EFTU both binds to EFTM with a similar binding affinity. In a complementary approach, we used mass spectroscopy to find whether the peptide alone, which is the N-terminal peptide 10-mer, can be methylated by EFTM, can be recognized by the EFTM or not. So the sample containing 10-mer peptide after incubation with EFTM and SAM were subjected to the mass spectroscopy. And as we see here, that the peptide alone and the peptide with EFTM, they show no change. So collectively, the gel filtration chromatography, the ITC, what we have done here, it shows that EFTM do have the ability to bind to the EFTU, which is even lacking the N-terminal sequence. On the other hand, the mass spectroscopy show that EFTM cannot methylate an isolated EF2 N-terminal peptide sequence. This points to the critical role for an extended surface of the globular body of EFTU in substrate recognition by the EFTM. We next generated a computational model of EFTM and EFTU using a protein-protein docking software, HEX. The final model was assessed based on the amino acid evolutionary conservation within the EFTM family, which revealed, as you show here in this figure, that the interacting surface is much more conserved compared to the solvent exposed surface in the in EFTM HM4. Here we show in yellow patches the important residues: arginine 81, aspartic acid 98, histidine 86, aspartic acid 91, aspartic acid 76 in the EFTM interaction surface, which are critical to interact with EFTU. And these are the residues which we later mutated to complementary charges just to disrupt the interaction and see how it affects the activity. Shown in the next figure is surface conservation of EFTM, as I was discussing earlier, and that the interacting surface shown here is much more conserved compared to the solvent exposed surface. This is an interesting thing also observed, you can observe here, is that the peptide channel where the N-terminal peptide goes into the EFTM is highly conserved, and most of the residues in the channel are at least more than 90% conserved in here. Now, to test our model, we generated mutants on EFTM and EFTU surface. The mutant EFTM proteins were expressed and purified as for the wild type protein, and their folding were assessed using thermal denaturation monitored by intrinsic fluorescence. We wanted to test the mutant proteins thermal stability because we do not want it to have results in which the activity is decreased due to thermal instability of the protein. So all of the mutant what we have tested are thermally stable, similar to the wild type EFTM. To measure the trimethylation of EFTU by the variant EFTM proteins, we used an established immunoblot assay with an anti-trimethyl lysine antibody. First, to determine the optimal reaction condition for comparison with wild type EFTM, a time course activity assay was performed, in which we used 0.25 micromolar of EFTM, the enzyme, and excess of EFTU substrate 10 micromolar, and excess of SAM co-substrate, that is 1 millimolar. And this experimental conditions revealed, as you show in the graph over here, that at 30 minutes, nearly at 30 minutes, the reaction is mostly complete. So we use this 30-minute time point for all subsequent methylation reactions. Collectively, the activities of EFTM variants with targeted substitution and the EFTU mutation with the corresponding changes experimentally validate the prediction of protein-protein interaction interface in our computational model. We next ask the question how EFTU N-terminal peptide might be positioned in the EFTM substrate binding channel. As you see in this figure, we used CAVR to determine the channel geometry near the SAM binding site, and also to determine the residues lining the substrate binding channel. In this case, as you see here, tyrosine 7, aspartic acid 15, methionine 74, tryptophan 170, and tryptophan 196. All of these residues are highly conserved, more than 90% conserved in the whole EFTM family. So we have a model also showing that the N-terminal peptide of EFTU comes in, and the K5 is positioned near the SAM binding site, and because there the methyl transfer is going to happen. And the succeeding residue, the phenylalanine 6 of EFTU, comes very close to the tryptophan 170, and we think that there may be an aromatic interaction which can hold it together. We tested all the mutants of the substrate binding channel, the M74, D15, Y7A, and all the other mutants show a very much decreased activity. In case of tryptophan 170 and tryptophan 196, it is totally depleted. The highly conserved EFTM residues, tryptophan 170 and 196, are positioned, as you can see over here, in the ends of the N-terminal binding channel, suggesting they may be performing a gatekeeping function. That means in case of 170, it comes very close to the phenylalanine 6 of the EFTU. That's how it can stabilize. On the other hand, the methionine, the first methionine of the EFTU, might be interacting with tryptophan 196. We next ask the question how the N-terminus of EFTU accesses the EFTM substrate binding channel. At least there are two distinct potential mechanisms which can be thought. The first, following the EFTM-EFTU interaction, the EFTU N-terminal sequence could enter via the adjacent opening onto the EFTM surface and thread into the substrate channel, as shown here in the figure, and then get fully accommodated. We call it a threading model. The other alternate mechanism would be binding to EFTU could induce conformational changes in EFTM that could open the peptide binding channel, allowing placement of the sequence directly into the final position, and this we call as the placement model. Now, given that EFTM can methylate N-terminal 6x histagged EFTU construct with the similar efficiency of a wild-type EFTM, the latter scenario, that is the placement scenario, seems to be more plausible. So we sought to further get evidence to support the placement model and to eliminate any potential alternative threading model. So what we did is we added a sumo domain at the N-terminal, which is the large domain, to the N-terminal of the EFTU. So if the threading model is true, then of course the sumo cannot enter through the small pocket. But if the placement model is right, then the EFTM pocket can just open up, and still the reaction can still take place, and the modification can still take place, maybe with a lesser efficiency. So we wanted to test it. So we first wanted to know whether the EFTM has an inherent ability to flexibly open the binding channel. We used a method called normal mode analysis, performed it on the EFTM. Normal mode analysis provides a coarse model of large-scale protein movements and can identify potential hinge points and the directionality of the domain movement. Doing this normal mode analysis, it suggests that the EFTM can undergo a large-scale conformational change between the methyltransferase core fold and the beta-5, beta-6 linker of the auxiliary domain. And these are the two regions which actually make the binding channel, that is the N-terminal binding channel. Now, to test the hypothesis, we added large sumo domain to the N-terminal of EFTU. Now, as we see in the Western blot over here, the sumo EFTU can still get methylated by the EFTM. That means there is no chance for the threading model, but only the placement model can work. We also generated two more mutants in which we extended the linker with 3 residue and 6 residue flexible glycine and serine rich residues, such that the distance between the sumo and the N-terminal increases. And what we see is that these two constructs show a further modest recovery of activity compared to the sumo EFTU construct. Collectively, the results support our model of placement of EFTU N-terminal into the widened peptide binding channel in EFTM upon binding to EFTU. Now, this is the final model of the EFTU recognition by the EFTM. What is shown here is that the EFTM binds to EFTU via an extended protein-protein interface, promoting conformational changes in EFTM that opens the peptide binding channel of the free form, and then it allows a direct placement of the EFTU N-terminal peptide. Within the substrate binding channel, the N-terminal peptide of EFTU is oriented by a group of highly conserved residues, which helps to position the lysine 5 for trimethylation. And this whole step actually goes twice and thrice to generate dimethyl and then trimethyl modification. We still are not sure whether it's a processive or a distributive mechanism. We are betting on a distributive mechanism, which it looks like to us, but further experimentation are needed to prove it. So, in a summary of all this, our analysis of EFTM-EFTU interaction evoke a model in which the protein-protein docking via an extended surface is required for the interaction of enzyme and substrate, as we have seen in the previous slides. There is a high residue conservation of the substrate recognition channel, where the EFTU N-terminal peptide is positioned for lysine 5 modification. And there is a role of protein conformational plasticity in the recognition and modification of EFTU. Now, there are other interesting things. Our current model also suggests that there is a mechanism of substrate recognition and modification is more complex in EFTU than the known bacterial lysine methyltransferases, which is studied, and also it is also different to the known set domain methyltransferases or the histone methyltransferases studied. EFTM is also very interesting because although it has the class 1 methyltransferase fold, it still resembles a lot of features with the set methyltransferase domain, which we need to investigate more. There are many more questions, including how the catalysis is done, how the EFTU is transported to the cell surface. These are all the questions which are under current investigation by our group. Overall, the work shows that is the first part in understanding the Pseudomonas adhesion with the epithelial cell, and this understanding of the whole pathway from a very molecular and from a structural point of view, it will help us to have design strategies or to inhibit EFTM and inhibit this adhesion to the host cell. I would like to thank Professor Graeme Conn, who is my mentor, Professor Joanna Goldberg, who is my co-mentor and collaborator, and the whole Conn lab, whoever worked in this project and otherwise, and the Goldberg lab. I'd like to especially thank Christine Dunham of Department of Biochemistry and her lab for her wonderful suggestion and advice in the project. I'd like to thank the funding agency, Cystic Fibrosis Foundation, for my postdoctoral fellowship, and NIH for the funding. And at last, I want to thank Rita for helping me making the webinar, its animation, editing, and the final production. So I'd be very happy to take any questions.